Hmm. All right, get myself all sorted. So welcome if you are joining me. If you have any questions you would like to cover, then please do feel free to ask away. If not, what I think I will do is I will just start going through some questions. Hi, Andreas, how are you? Have you got any questions you want me to cover tonight specifically? I'm good, thank you. Although I have to say, I'm starting to get a bit of a scratchy throat, so I'm wondering if I'm about to get something. Hi, Chidima, how are you? I see you've been watching my YouTube videos. I keep seeing your comments popping up. Unless there's someone else who has the same name as you. Is there anything you want to cover before tomorrow then? Have you got a specific topic area? You're an OCRB, so of course not OCRA, but do you have advice on how I can revise for the practical skills? Because I'm always get confused when it's random experiments. So, okay, do we need to know about agglutination? Yes, you do, Andreas. You do need to know about agglutination. So if you want to know with regards to practical skills, one of the key things that I would say when it comes to anything to do with practical skills, so let me just get up a little bit here. So for one of the questions that I found quite interesting in the 2024 exams is they were pushing more on practical skills in the AS papers, and in the A2 papers, they still ask the same sorts of questions, but just not in the same details. So let me just bring in my module 1 book. So if we look then at the practical skills, I'm going to bring this down here a second, and I will switch screens and share. So one of the things when it comes to practicals, so this is my practical study guide that I did on development of the practical skills that will be assessed. So the first thing I would say to you is experimental design. What you need to know about experimental design is to do with the fact that you need to know what your independent variable is, you need to know what your dependent variable is, and you need to know what control variables there are. So whenever you are given a practical, whatever that practical may be, the first thing I would always suggest you do is think to yourself, right, well, what is the independent variable of the practical that they have given me? What is the dependent variable? What am I measuring? So for example, in this case, I've said that the independent variable is the concentration of the substrate with regards to hydrogen peroxide. So if I was going to do this and I was answering a question about this, then I would need to say that my independent variable is the hydrogen peroxide concentration. I want to give 5 values. So if I'm going hydrogen peroxide concentration, I might put 0, no hydrogen peroxide, but then you'd be looking at a 2%, a 4%, a 6%, etc., which would be you giving 5 values for a concentration of the substrate. So that concentration of the substrate is what I'm changing. Therefore, I need to control things like the volume. So if you look down at my list that I have here, my control variables, these control variables can be applied to any of the questions that you have. So if, for example, they're changing the substrate concentration, you want to control the enzyme concentration, the volume of the solution, you want to control the mass. In this case, they're looking at liver. So it could be that you're doing 5 grams of liver. If you're looking at potato and you're looking at catalase, you want to control the surface area that's exposed to it. Things like that are going to help you to be able to answer whatever question they give you. So whenever you've got a practical, always, what's the independent variable? What's the dependent variable? What are your control variables? Have at least 3 control variables. And then what you then want to do is say, okay, obviously, first thing, if it's a rate question, make sure you're doing it over a fixed length of time. Make sure as well that your answers are reasonable to the question. So have a look for that information. If they haven't told you it in the stem of the question, then you need to say that hasn't been controlled. So therefore, I would need to improve that if I'm going to do the practical again. The other thing to note is a control variable is different to a control. A control is something that you're going to use, same experiment, but this time it's going to be missing the independent variables. So you're not changing whatever it is that you're looking at so that you can compare what would happen naturally if I didn't have a different hydrogen peroxide concentration. So for example, what would happen with the liver cells if they were just put in water? Would these oxygen bubbles be released? And then I'd be looking at comparing that to when I put the hydrogen peroxide in. So you can either do it without the enzymes or you can do it without the substrate, depending on what it is you're actually wanting to look at in the investigation. Obviously, repeats 5 times, and then obviously you count a mean, a standard deviation, and do comparison. Now, one of the things that I've noticed is students, when they do the comparisons of the statistical test, what they need to understand is, if I just skim through, make sure you know your statistical test. So do you know how to do, for example, an unpaired t-test? Do you know how to do a paired t-test? These are the sorts of things that they will be asking you to do in the exam. Right, Chidima, you have asked for the cardiac cycle. So I'm going to jump back over here and do a little bit on the cardiac cycle. I do have a feeling, you're very welcome, that there's going to be something that's linking the cardiac cycle with an ECG. They haven't done very much on that, and also linking it potentially to the sympathetic nervous system or parasympathetic. So yeah, the practical skills are all about you having that normal level that you can go to, have your go-to answers, your flashcards, etc., based around that content, and then build from there. Hi, Joyce. Right, so where am I going? I want to do on the cardiac cycle. Do give me 1 second. I was actually looking at the cardiac cycle in my group tuition session the other day because I really do feel that neuronal communication, muscles, now it's whether or not they're going to ask you to compare the smooth muscle, cardiac muscle, or involuntary muscle, cardiac muscle, and the, what's the other one? My brain's not functioning. You can tell I've been teaching too much. Oh my word, skeletal muscle. Why could I not think of that word there? So yeah, so it's one of those things that I believe they're going to bring in those different sections. So if I just do this thing, rub out my answers. So if you were to look at, for example, a diagram that looks like this. Oh, well done. I'm glad that it went well for you. Is AQA easier than OCR? AQA is just one exam board that's the styling of questions are slightly different. You've got your paper 3, which is the 25 marker, which is difficult for some students who can't do the long response questions, or it's the OCR where you only have 6 marks in your papers, but you've got more applications. So it just really depends. The content's still the same, to the same level because it's a level 3 qualification. So Chidima, you said you don't know how to answer this question. So can anyone who's in here, fight or flight? I do feel like the fight or flight response could come up, but I think it will be more linked to adrenaline and hormonal activation potentially. Iris, hi, Iris, you are telling me that we've got the bundle of his. Now be careful. So A is your sinoatrial node. B is your atrioventricular node. So be careful on that one. Then you want to look at your C, which is your bundle of his. So you did have the right name, but just in the wrong position. And then your D is your Perkine. Oh, I'm glad you found me on my YouTube channel. Perkine fibers. So when you're looking at this, that's what you want to be able to see. That do you know the different parts to it? Can you actually label these? So then what we want to do is what I want to show you. She says, just rub out my bits I was doing previously, is what a question might look like. Now, some of these questions are AS questions and some of these questions are A level, but remember what they will be doing is they will be assessing you on your A level content, but this is still part of the A level, because if they're looking at the cardiac cycle, that's module 3, which can come in paper 1 or can come in paper 3. So they can bring it in whichever way they want. I do feel there'll be an application question, but this is definitely more a AS question if we look at it. And this one is then asking you to apply your knowledge to what they've given you. Now, a lot of students are able to note that this part of the graph is showing you, as it says, the left atrium. I love highlighting. In the exam, I would always suggest to do some highlighting, because when you highlight, it actually helps you to visualize what the graph is showing you. So my ventricular pressure is the green here, and I'm going to show this so that we can see that is slightly higher than the next one, which is my aortic pressure. So by using highlighters in your exam, it can help you visualize what's actually happening so that when you're looking at it, you can separate out each of the different parts. So here we have at A. So at this first point down here, at A, it says the muscles in the wall of the atrium are contracting. We can see that there is a contraction because we can see there's an increase in the pressure, and then there's a decrease in the pressure as well. So again, if they're giving you a graph, and this comes back to practical skills, some of your practical skills that you'll be assessed on, they will actually be asking you to analyze graphs. So this is a graph, and so therefore we need to note that there is times. So they've told us down here that the times are in seconds. And also what we can see is each of these 0.3, 0.5, 0.7, 0.1. So use the scales to help you note where things are happening. If they are asking, so in the past, they've asked a question for students to describe and explain what is happening in the graph. So make sure you're actually giving a time period from 0 to 0.13, let's say, seconds. There is an increase in pressure and then a decrease in pressure because the atria are contracting. So that's what you want to be explaining, that describe the graph, explain as well. Now, this isn't answering this question. Remember, I never just answer the questions that are in front of me because they're never going to give it to you in exactly the same way. What they're going to do is they're going to assess that knowledge in whatever the way they want to. So they'll use the same graphs again, but they'll just ask different questions relating to that. So make sure you do give timings. It's going up, it's going down, for example. So this one says, this is caused by a wave of electrical excitation that starts the atrioventricular node. Well, that's not correct. It's the sinoatrial node. Absolutely. It's not the AVN, it's the SEN. So that's the first thing that's incorrect in this question. At B, so now we're down here. So we're at that point there. At B, the muscles in the wall of the ventricle are contracting. So what we're seeing is ventricular, excuse me, systole is occurring. So the contraction of the ventricles are happening. The atrioventricular valves are open. Now, this is an interesting one because some students get confused about those valves and where they are and what they are doing. So let me just bring in, and I have seen your message, and I saw it on TikTok as well, about communicable diseases. I will jump to that after I have done this for Chidima. I want to go to this one. I want. Yes. Okay. What I want to do is, I'll come back to this question in a second. I want to look here. Now, it's important whenever you are describing to do with the pressure changes in the heart that you understand it's about pressure from one place to another. So in this first diagram, we call this number 1, what we can see is our pressure is higher in the atria than the ventricle, and therefore that is resulting in the valve being opened. So if there is a higher pressure in the atria, that is going to force the valves open, and it will do that because of that atrial contraction. So atrial systole. Is it true that the heart would explode if the ventricles and atria contracted at the same time? So if the atria and the ventricles were to contract at the same time, it wouldn't explode. What would happen is you would have a pressure difference that would cause a problem. So what you've got is when the pressure gets greater down in the ventricle compared to in the aorta, in the atria, what's going to happen is that closes the valve. So what you have is the valves, the atrioventricular valves, work like swing doors. They will swing down and open, but when the blood fills up from underneath, it closes them to that point where they're no longer able to flip backwards. So they shouldn't be flipping backwards because you have got the connective parts, what we call the heartstrings, which are really strong. And that ensures that the valves actually open in this way when the pressure is greater above, but when the pressure is greater below, that should actually close the valves. So therefore, our atrioventricular valves would be closed. So this one here is our atrioventricular valve. So our atrioventricular valve is going to be open when the pressure in the atria is greater than the pressure in the ventricle. The atrioventricular valve is going to be closed when the pressure in the ventricle is greater than the aorta. So, oh, I keep saying aorta, the atrium. See, you can tell my brain is tired. This is like my 8th hour. Apical dominance. Yes, I will, Joyce, I'll do that after I have looked a little bit at communicable diseases. So let me just finish this one first, and then I'll move on to communicable diseases, and then I'll jump back to apical dominance. So if we look here, then we're told we're at this point here. Now we can see with the green that the ventricular pressure is going up now. The ventricular pressure is going to be greater than the atrial pressure. And because of that, what's going to happen is that atrioventricular valve is not going to be open. It's therefore going to be closed. So that's an important part to note. And the pressure obviously in the aorta is an interesting one as well, because this says the pressure in the aorta falls. Well, if we look at the green, as the green is going up, 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 if we then look at the pink, what you can see is the pink/purple is going to also increase. So that's another incorrect statement that they've said here. It doesn't fall, it rises. It rises along with the ventricular pressure. However, it is lower than the ventricular pressure. So they can use the term tricuspid valve or bicuspid valve. However, what they've done with the A level is they've made it easier for them in that they just need to say atrioventricular valve. So they've removed that, I wouldn't even say it's complexity of it, but they have removed the tricuspid, bicuspid, and just said, call it the atrioventricular valves. So the mitral valve is just basically another name for the bicuspid valve. So again, that's just an atrioventricular valve. It's just another name for it. So just to go back, I'm going to change colors here and talk about the pressure. If I look at picture number 1 again, I have got a higher pressure in my aorta or pulmonary artery, depending on where I'm looking. So don't forget, our atria is on both sides and our ventricles on both sides. So therefore, higher pressure in the atria, lower pressure in the ventricle. When the semilunar valves are closed, that's because there is a lower pressure. So these are my semilunar valves. There is a higher pressure in the artery than there is in the ventricle. So when we look back at, oh no, I went too far, back at the question, and then I'll bring that one in. So when we look back at the question, we can see the ventricular pressure here is lower than the aortic pressure. Therefore, only when that green line goes above the pink line, so the pressure in the ventricle gets higher than the pressure in the aorta, will my semilunar valve open. So as we carry on, we can see that the ventricular pressure is increasing and then decreasing again. When the ventricle pressure gets below the aortic pressure, that's when at point C, my semilunar valve is going to close now because the pressure is greater above than it is. So going back here, we have a higher pressure, a lower pressure, higher pressure, lower pressure. So again, it's all about where there is a greater pressure. So it's really important that you note the difference between the pressure in each of the different chambers. So when we're talking about the atria to the ventricle, or whether we're talking about the ventricle to the arteries, which are going to carry the blood away, and it's about these valves that can open and close. It's not a mistake. There's just a lot of content to learn when you are doing the course, basically. So here we can see there is a lower pressure up here. There's a higher pressure down here. That's why this valve is closed. This valve is also closed because there's a higher pressure and a lower pressure. If I have a higher pressure and a lower pressure when I compare my ventricle to my aorta or my pulmonary artery, that is when we will have those semilunar valves open. So it is important that we note, as we go back to that question, where we're at and the difference in the pressure so that we can tell which of the valves are open or which of the valves are closed. So as it comes back down, as the ventricular pressure drops back down, when we get to the point which is here, which isn't labeled, we'll call this D, we can see our ventricular pressure has now dropped below the atrial pressure. So now my atrioventricular valve is going to be open and blood is going to be flowing passively at this point into the ventricles from the atria. So again, like I said before, make sure you're using the time periods to show that is where it is passively flowing from the atria to the ventricle, because the atrioventricular valve is open. Between here and here is where we had the atrial systole, the atria contracting, and therefore that is going to be an increase and a decrease, but we can't see that increase and decrease yet. That's going to come over here, and then we'll see it again. So why is there a slight increase? That's an interesting one, Beatrice. So what you have with the aorta, remember, if I go back to my diagram a second, let me just bring this one back in. So you can see the semilunar valves are like little pockets. So what happens is those pockets, actually, if I try and get this to show you, imagine that's the wall of the artery and this is the little pocket. What happens is this little pocket lies flat, and then when the blood starts to fill, the pocket will actually do this. Now that's obviously on both sides. So you end up with this filling, this coming together. So normally what I do is I'd have like 3 beakers and I pull them together. When there is a full pocket, so as you can see here, this pocket is completely filled with the blue color, and this one's completely filled with the purple color. What happens is there's a slight push down. So once it fills and those pockets and the valve shuts, there's a little bit of a push down, and that little push down is that little blip you get where there is an increase in the aortic pressure. So that's because once the valve's closed, there's that little bit of a push, and that push gives that little blip that goes up with regards to the pressure. So I hope that helps. The other thing I would say is just to note, because I do feel like the cardiac muscle is coming in if you are in year 13 as well as year 12. Striated muscles, I do believe skeletal muscles are coming in, and the sliding filament theory. How they ask the question, no idea. But it is expected that you understand about the sliding filament theory. And I do think that the neuromuscular junction and how that then activates the release of the calcium ions will be interconnected. Anyway, striated muscles, if we're looking at the cardiac muscle and we're looking at the heart, we need to make sure that we understand that 1, it's striated, so it's striped, it's got a striped appearance. 2, it's branched, because you can quite clearly see in my diagram that I have these branches to it, and is uninucleated, and therefore that means it's got 1 nucleus per cell. So uninucleated, 1 nucleus per cell, so we can tell that one, the cardiac muscle, from the skeletal muscle, because the skeletal muscle is multinucleated. And the other thing to note is, again, the cardiac muscle is able to contract together as a synchronized unit, and therefore it has these connections between the cells, and those connections are called intercalated discs. Now, they would give you that information in the exam, in the stem of the question. They don't expect you to know that they're called intercalated disc. They do expect you to know that it's branched. Now, what's important is those intercalated discs are actually composed of cell membrane proteins. And those cell membrane proteins actually connect the cells together. And that means that an action potential, the movement of the sodium ions, the depolarization of the cells can happen together rapidly as one. So I definitely feel neuronal communications and muscles are coming in. So if you are year 13, make sure you are confident with the details, i.e. where's there a higher concentration, where's there a lower concentration, which ions are moving in which direction, and what does it mean when it says an action potential? So have your keywords there. Right. Maybe a transverse section of a part of, yeah, I do. It could be they've only done that once. One question, which was of the sarcomere, excuse me. And I do feel that it would be something that potentially could come up, or looking at the proteins and linking it to maybe mutations in the proteins that means that the muscle doesn't contract in the correct way. Who knows? I'm just making it up on the spot here, but that would be potentially something that could come up. If you're still in here, when you want to look at communicable diseases, which part of communicable diseases are you looking for? Could you go through label and dissection of the heart? I mean, a dissection of the heart literally will just be draw what you see. You've got to draw whatever they give you as the picture. So if, for example, you can see my heart at the top. Yes, I do. Sorry, Lois, I do. I do feel like that one is going to come up as well. And I've got a feeling that they're going to bring in, let me think this one through now. I definitely feel like regulation of genes is coming in, and I feel like they're going to bring in genetic engineering and maybe somatic cell nuclear transfer or embryo twinning would be my thought. You're welcome, Chinema. Just to go back on how to do the dissection, they would literally want you to draw, this is a really bad drawing. See, something like that looks nothing like the picture above. So you need to make sure that your proportions, you don't have to be an artist to be able to do this, but you do need to draw what you can see in the picture. So if they give you a diagram to copy, I would say that you want to just draw it whatever you see, and then they would be expecting you to draw little bits coming down. So you see one that's coming down here, lines coming off. If they were going to ask you to do that, if they're asking you to draw the heart, I would start off with my pencil, drawing a box, and then what I would do is I would then add in my extra bits to bring in my septum. I don't think they'd ask you to do that. I do think a diagram is coming in on a biological drawing, but I don't know what it will be. Okay, so I'm not sure if you are still here. So I'll jump to Aplasimo's pathway predictions for you. Right, let me jump to the apical dominance first. Let me do that one. Where do I want to go? I want to go to plant response. Bear with me, guys. My brain is on slow go today. This is my 8th hour of teaching today. So I want apical dominance, which therefore means I want it in this one. Don't I? Yes. Here we go. No, I don't want that because I've changed that. I updated it. Where did I update it? For next week grip tuition session. That's what I've updated it for. Where are you? There we go. This is the thing. Everything I do, I am always adapting it. Always adapting the stuff that I do. Okay, so apical dominance then. I'll do that one. No, definitely no espresso. I need to be able to sleep. My girls are not sleeping very well, so it's also made me a bit tired. Apical dominance, what you should know is you have apical meristems. So we know that we have these undifferentiated cells, which are able to go through mitotic division and then differentiate. So if we have what's known as our apical meristems, these are found at the tip of the roots and the tip of the shoots. So this is important, knowing where it is. Then what you have to understand is that the plant hormones are used to control growth. So hormones can work antagonistically, and that's important for you to note, because of their concentration. So a plant's hormone is going to be a hormone that is produced somewhere, released into the or released out of the place where it's made, and then able to move throughout the plant. Now, whether that's through the transport system of the plant or whether that's just through diffusion through the cells depends on what the question is that they're asking you. So the important thing we're going to talk about for apical dominance is auxin concentration. So you should know that auxin acts as a growth promoter. You should know that it controls cell elongation, maintains the apical dominance, and is also involved in tropisms. So if we were looking at what auxin actually does, you should know that auxin is produced at the apical meristem cells. It is released. It will then diffuse down through the plant. And what it will do is when it gets there, it is going to alter gene expression. So remember when I said before that I believe the regulation of genes is going to come in? They could link it to plants. So auxin alters gene expression. It's a hormone that's going to bind to a receptor. When it binds to a receptor, what it actually does is it causes the increased production of a proton pump. So auxin is going to bind to a transcription factor inside the plant cell. That's going to increase the production of these proton pumps, which will be inserted into the plasma membrane. And what that will then do is that will then cause hydrogen ions to be actively pumped from the cytoplasm into the cell wall. So they expect you to know that the cell wall is made up of cellulose. Because it's made up of cellulose, it's got your beta glucose molecules. They are 180 inversion every subunit, and that therefore results in these cross links between the fibers, the cellular fibers. So we need to understand that. So we've got hydrogen ions being pumped in. Those hydrogen ions are going to decrease the pH. This is going to activate enzymes, and those enzymes are called expansins. Now, if they give you a question, there will be information about the fact that it's called expansins and the fact that it's an enzyme. What they're wanting you to note, it's an enzyme, so therefore it's a globular protein. It's a globular protein which will change its tertiary structure because of the decrease in pH, because remember, your globular proteins are pH sensitive. So this is where module 2 comes in, and you understanding enzymes, but they could hide it behind talking about auxins and looking at how that hormone is going to affect the cell elongation. Excuse me. So what you then need to know is, because we have this pH that's decreased, because these enzymes have now been activated, we have post-translational modification occurring in the fact that it's been changed. we are going to break our cross bridges. This is OCRA, breaking the cross bridges between those cellular fibers. That's going to make the cell wall more flexible. If it makes it more flexible, more water can actually be absorbed. That's therefore going to result in the turgor pressure pushing the walls outward. The cell expands, and as the cell is expanding, that is elongating the cell. So when we talk about promoting cell growth, that's what we're talking about. The auxin is going to promote the cell growing by allowing more water in, by making the cell wall more flexible. So how does that link to apical dominance? Well, that links to apical dominance because what you should know is it's all to do with concentration. So if you have high auxin concentration, that is going to stimulate the growth at the apical shoot. Well, remember that the auxin is going to diffuse from the apical dominant, from the apical meristem down through the stem. Well, what you've got is it's going to be at a high concentration up at the apical bud, therefore ensuring that the plant is going to grow upwards. What you have is if you have a high enough concentration, that is going to inhibit the lateral bud. However, the further away we get those lateral buds from the apical meristem, the concentration is now going to decrease. So what you'll find is that the apical dominance is ensuring that the plant is going to grow taller. Growing taller means it's going to be able to be exposed to more sunlight, therefore more photosynthesis occurring because there's more light dependent reaction. And then you have that we are going to then use that to produce the necessary carbon compounds that can then be used for the production of ATP for all the metabolic demanding processes within the plant. So if we understand that and we look at, and this is a graph that they have given before, where there was a stimulation of growth percentage, inhibition of growth down here. So the key thing to do, if it were me in the exam, is I would be using my highlighters to highlight the different parts that they've shown me with a key. So what I can see here is my blue goes up and then back down again. My yellow goes up and then it goes down again. So they both go up and they go down. But what's really important is it's zero growth. Anything above here, they've said, is a stimulation of growth. Anything below here is that inhibition of growth. So now I want to be using my numbers. So anything from this point to this point here, probably in between here, we have got a stimulation of the lateral shoot. So when the concentration of auxin is low enough, then there is a stimulation of growth in the lateral shoots. Whereas if the concentration of auxin gets too high from this point onwards, then what we have is that you can quite clearly see past this point here, we are now inhibiting that lateral shoot growth. And therefore, that is where apical dominance is being shown in that graph. So I hope that helps on the apical dominance side of things. Right, so where are we at? I'm guessing A is gone. I'm not sure if you're still here. Canceling homeobox and Hox genes, you mean? AM, I will bring that in a minute. I'm going to do. Could you please go over specific and non-specific response to pathogens in the body, excretion? You're welcome. Guys, if you are just coming on here and finding my videos, if you are doing OCRA and you want specific videos on this content, you've got a topic area, you should be able to go to my playlist for that module and watch a video specific for that topic area. So if I don't manage to do what you're looking for tonight, go and check out my video that's already on my YouTube channel. The whole course is there for you to use and to access. What was I doing? I was going to do Apoplast and Symplast pathway. Chidima, are you still in here? I'm not sure if you are still here or not. No, I don't think you are. So I won't do that one then. What did I want? Apoplast and Symplast pathway. I'll start with it with regards to roots. And then I'll move on to it with regards to loading. Oh, and another question could be on CR dilutions for that one. Okay, so I'm sending links to students to all your videos. Oh, thank you. Right, who was it that asked for it? Your mum's life. This is for you. Okay, so what have you got? Symplast pathway, Apoplast pathway. So I've used the different colors with my arrows to represent the Symplast pathway and that Apoplast pathway. So here you can see that the Symplast pathway, the red arrows are going into the cells and then making their way through these gaps within the connections between the cells. So what's important for you to note is the Symplast pathway is continuous cytoplasm. I love my alphabet and I use it to help me to remember. Symplast pathway has got a Y and therefore cytoplasm also has a Y. That is my continuous cytoplasm connected by my plasmodesmata. Now, this is important for you to remember. For the water to get through, the water has to pass the bilayer. So therefore, the water is going to move in by osmosis. It's going to be a net movement from a high water potential to a low water potential. And it's going to go across a partially permeable membrane. So that's important that we note that the Symplast pathway is that the movement is getting into the cytoplasm, so across the cell membrane, and then it's going to make its way through as a continuation of the cytoplasm all the way through the cells, through the epidermis, through the endodermis, to the xylem, and then up through the plant and wherever it needs to go. So that's important for you to note. Oh, this might not actually work in a minute. It's deciding it's being a bit annoying. But I'll keep going. I might not add my notes. If we go the Apoplast pathway, this time we're looking at that cell wall. So remember that cell wall, oh, it is doing it good. The cell wall has the cellulose polysaccharide. So because it's saccharide, because it's made up of the cellulose polysaccharide, what do we know? That cellulose polysaccharide is insoluble, so therefore it's not going to affect the water potential. So what we have instead with the Apoplast pathway is that water is going to fill the spaces between the cellulose fibers. And we know that water has cohesive property in the fact that they will form hydrogen bonds between the water molecules. So if we go, water is made up of 2 hydrogens and 1 oxygen, delta positive, delta positive. Do not forget your delta signs and a delta negative. And if I have another water molecule over here, I've got my delta positive sign, delta positive, delta negative. I know I've got hydrogen bonds that are going to form between because of that cohesive properties. Therefore, what's going to happen is mass flow of water carrying any of the substances that it has collected along the way is going to be transported through the apoplast pathway. Paul, do students have to link this using keywords like the transpirational pull? Yes, if we were talking about transpiration, I've not gone there yet. I just want to talk about the symplast and apoplast pathway, but yes, absolutely. They have to know that because of transpiration, that evaporation of the water from the mesophyll layer and then that diffusion of the water vapor out through the stomata results in this pulling up because of the adhesive and cohesive properties of water. If you are AQA doing it, you just need to know it about the tension. What's it called? Oh my gosh, my brain's not going into it. They don't need to know about adhesion. They just need to know it. It's called the tension. There's another word that's missing out my brain, but yeah. Cohesion tension theory. Thank you, Iris. That's it. The cohesion tension theory. That's why I was like, why is it not fitting together? Because the cohesion is to do with obviously your water to water, but then you've got that tension as well. Okay, these parts here, these dark sections are important in knowing that these are what's called the Casparian strip. So this is a band of waxy material called suberin. It's a waterproof layer. And what that does is that stops the apoplast pathway continuing on into the rest of the plant. So it diverts the apoplast pathway into becoming the symplast pathway. So the membrane is going to stop any of the substances that it doesn't want to get into the cells or into the plant. By having this Casparian strip, it is a mechanism that actually helps the plant keep things out that are too big to get across the bilayer, that are charged particles that don't have the right channels to get through. So therefore, it is a way in which the cells have differentiated so that they are stopping the entry, for example, of a pathogen. So that's an interesting one where the Casparian strip is that band of waxy material repelling the water, diverting it into the symplast pathway. Okay, I'll probably do about another 10 minutes. Where am I at? Where did I say I would do? I hope that helps on that one for your test tomorrow. I don't feel like this will be a wrong thing for me to say. I don't feel like excretion in the kidneys is coming up. I feel like it's potentially more going to be linked to animal responses and there'll be more multiple choice questions. There's been a lot of excretion connections in the last 2 years, but I could be wrong. Okay, where do we get to? It's hard to know what's going to come up. I've done a lot of research. I've done a lot of thoughts. But on when did I about what comes up? But I do. Oh, homobox genes. Thank you, Paul. What would I do without you? Where did I put it? Give me a second. I did make a little mini prediction when I did my Easter course. Or I wrote a list down. I can't remember where I put the list though. I feel like the cardiac. Oh, there we go. Hold on. If I were to make any predictions whatsoever, because we're getting so close to the exam, then this would be my predictions. So my predictions would be batch and continuous fermentation, immobilized enzymes, muscle. I feel like gibberellins and germination. natural selection neurons, your brain, the nitrogen and cardiac cycle, lipid-based hormones. I really feel like lipids is coming up huge because they did so much on polysaccharides in the last couple of years that I feel like the lipids, because you can go in so many different directions with lipids. So your phospholipids, lipids as an energy storage, lipids entering the respiration at different parts because of your spiritual quotients. Percentage efficiency with trophic levels, they have done very little in that lately. I definitely think you're going to do a line graph or a histogram. I do feel like you're going to either get a T-test or a Spearman's rank because they did chi-squared the last 2 years. They've done a lot of chi-squared. I feel like it's not actually listed on here, but fisheries haven't come up very much either. Evolution, inheritance, insects, and animal cloning. That would be my kind of ones. No, medicine with silver is not just for paper 1. So batch and continuous fermentation is paper 2 or paper 3, as is immobilized enzymes. Muscles, germination, paper 1. Natural selection, paper 2 or 3. Neurons, paper 1. Brain, paper 1. Nitrogen cycle, paper 2 and 3. So remember, 3 is any of them. Lipid-based could be anywhere because it's module 2. DNA replication, module 2. Differentiation, module 2. Gene, paper 2. Percentage efficiency, paper 2. Line graphs, anywhere. Plant disease and defense is paper 2. Animal cloning, paper 2. Insects, paper 1. Inheritance, paper 2. Evolution, paper 2. That would be what I would kind of say based on what they haven't assessed lately. I feel like translocation, but I feel like translocation in a practical sense or respiration in the practical assessment. I feel like you've got a biological drawing coming up. And I feel like it could be linked to mitosis, but again, I don't know. So yeah, that's where I would do. Yeah, all my videos, basically every single module, every single content video is out there already. I am going to, unfortunately, I don't know if I'll have time to do it for you guys who are sitting the exam now. But I am going to do more videos on the practical side of things. But you'll notice I've been putting more and more of these lives out there to help you in how to answer the questions. But as I say, for those of you who come to my lives quite often, I have to prioritize what actually brings the money in for me. I don't get paid for this. And so therefore, the priority goes to me doing those sessions. I do a live on a Wednesday, every Wednesday, and it depends if it's a Monday or a Tuesday, because I've moved to Tuesday because of my group tuition sessions. It tends to be more a Tuesday now. So like I did one last night. But the good thing is because I do them on my YouTube and I do them through Zoom, they are automatically live streaming for you. You can join the live stream on YouTube, or you can join the actual Zoom because I always put the link in there. But the other thing is it's saved for you to have access to afterwards. Whereas when I was doing it on TikTok, unfortunately, I had to download it and it took about an hour and a half to download each video because they were so long, because you know me, I just chat away. And so therefore it was taking too long to do that, which is why I moved over to do it this way. I am going to be offering group tuition sessions for new year 12s and for year 12s going into year 13 from September. I do run group tuition sessions now, and I'm offering a course again in May half term, a 2-day intensive course for anyone who is obviously looking for a intensive way of doing that revision. You can always book on to my course, which is 2 days from 9:30 to 4:30 on the Wednesday and Thursday of half term. And that is working out about £25 an hour, which is the cheapest I can do. But I am, guys, going to stop for tonight. I will be back on, if not Monday, on Tuesday. I do pop on if I have any time that I can offer. But when I do these sessions, these are more a Monday, Tuesday / definitely Wednesday. So thank you for everybody who has been with me tonight. Thank you for joining me, for watching my videos, and yeah, keep sharing, keep liking. I want to make more, I want to do more. And so yeah, thank you, because obviously the more you guys turn up to these sessions, the more it means that they get pushed out to other people. So thank you and good night.